All right. Hello, everyone, and welcome to today's Cryo-EM Current Practices webinar. You're joining the three NIH-funded National Cryo-Electron Microscopy Centers. My name is Christina Zamani. I'm a scientist and training liaison at the National Center for Cryo-EM Access and Training, NCCAT. I'm joined today by staff members from each center, including my colleague here at NCCAT, Ed Eng. Joining from the Stanford Slack Cryo-EM Center are Nathan Burrows and Michael Schmidt. And from the Pacific Northwest Cryo-EM Center is Maurizia Mileto. Before we give the floor to our speaker, we'll have a quick update from each center on the resources that we make available at no cost to the research community. For our main event today, PNCC has invited today's speaker, Professor Sarah Clark, who's joining us from Oregon State University. And Maurizia will introduce her properly after our center updates. This webinar is a regular event we host every month, highlighting users of the National Cryo-EM Centers who generously share the tips and tricks that have enabled them to make use of cryo-EM in their research. You can save the date for our talk on October 30th. Evan Wood from San Diego State University will be giving a talk on overcoming complex dissociation, cryo-EM structure of 7-diazopyridine in DNA proteins A and B reveal structural basis for DNA sequence recognition. I think he'll have an interesting story about how to deal with things that like to fall apart when you put them on grids. A registration link is already on the events page at cryoemcenters.org, where you can also find recordings of our previous talks. For final logistics today, if you have questions about accessing National Center resources, please feel free to use the chat to reach out directly to any of the panelists here. And for questions for our speaker during the talk, please use the Q&A box, and Maurizia will moderate Q&A at the end of the talk. So let's go ahead and get started. First, I will turn it over to Ed to let us know what's new at NCCAT.
Hi, I'm Ed here at NCCAT. We offer data collection on one of our 5 Krios instruments, grid preparation and screening on our blot-free chameleon device or a plunge freezer, and access to 2 Glacios instruments, pardon me. And we also offer training. On the training front, next week, we'll be having a symposium on integrative structural biology. Even if you haven't gone to the symposium or haven't registered for that, we will have a companion workshop that still is open for registration, including tomography workflows with the Arctis, data collection using the Alpine and Latitude, as well as the SynCell microscope, which is good for precision photo labeling. So if you're interested in learning a little bit more about the workshops happening next Friday and you're around New York, you can always email us. Also, something that's going to be happening next spring is our single particle short course. Registration and applications for that will open up later this month. To find out more, go to nccat.nyspc.org.
Thank you, Ed. And next, I will turn it over to Nathan.
Good morning. At S2C2, we have the same suite of access programs that most of the centers have, from high-end data collection to assistance with optimizing your grid preparation, as well as training programs where we teach the entire pipeline workflow for single particle ET and micro-ED. What I want to highlight today for everyone is that we are having a 3-day workshop on structure modeling into cryo-EM data and the structure validation. Unfortunately, registration has already closed for this workshop, but we will be posting recordings of the workshop onto our website afterwards. And we hope you can at least take advantage of those.
Thank you, Nathan. And Maurizia, let us know what's new at PNCC.
Good morning, everyone. My name is Marzia Miletto, and I'm a microscopist at the Pacific Northwest Center for Cryo-EM in Portland, Oregon. And I would like to briefly talk about what we offer here at PNCC. We have 2 types of proposals: screening access, which allows you to come on site for up to 5 days of sample prep and screening with the help of staff. And these proposals is mainly aimed at low resource institutions. And we have another type of proposal is general access, which awards up to 480 hours of microscope time a year for 2 years. This time can be used for screening or data collection. All approved general access proposals are also eligible to participate to our training programs, and we offer a variety of workshops that cover the entire cryo-EM pipeline from grid prep to screening, data collection, data processing. And also all approved proposals are assigned a PNCC staff microscopist as a scientific point of contact or SPOC. This person can help you with questions about sample prep optimizations or strategies for data collection and so on. In terms of resources, we have 6 cryo microscopes, one Arctica, a Glacius 2, and 4 Crios. We have one of our Crios is equipped with a Falcon 4i detector and a Selectris X energy filter, one with a K3 and a Biocontinuum HD energy filter, and we have 2 of these Crios with both a K3 and a Biocontinuum and a Falcon 4i with or without Selectris. We also have VitraBots like a GP2, VitraJet, manual plungers. So if you're interested in working with us, please go to pncc.labworks.org and apply. And our deadline for proposal review is the 1st of every month. And you will find all the information on our website, but please reach out with any questions. And we look forward to hearing from you and see your applications. And that said, I'm so pleased to introduce you today's speaker, Sarah Clark. Sarah received her PhD from Oregon State University in molecular and cellular biology, and she worked as a postdoc researcher at Oregon Health and Science University, and is now back at Oregon State as an assistant professor in the Department of Biochemistry and Biophysics. So the focus of her research is on the structural biology of membrane proteins, and in particular, she's interested in understanding the molecular mechanism of lipid transport, as well as of sensory transduction, using a range of advanced techniques such as cryo-EM. And in May 2025, the Clark Lab published a groundbreaking paper in Nature, providing fundamental insight into how cells transport lipids and maintain lipid homeostasis. And this process is essential for brain function, metabolism, and immunity. And they determine the structure of a major lipid transport protein and discovered 2 key helper proteins. So by showing how this protein work, Sarah's research helps understand how a faulty lipid transport is linked to disorders, and this opens the door to the design of new treatments. And today she will be sharing her knowledge on this topic with a great talk on the architecture of a bridge-like lipid transfer protein. And I think you will find her perspective both fascinating and inspiring. So please join me in giving a warm welcome to Sarah. And please, Sarah, take it away.
Awesome. Thanks so much for the introduction. That was great. And thank you all for coming today. I'm excited to talk about our work on bridge-like lipid transport proteins, otherwise known as BLTPs. And I like this picture in the background here, which shows a bridge over a very large body of water. And you can see large transport trucks moving across the bridge because it's sort of analogous to lipid transport in a cell. In a cell, lipids are made in one location and then have to get transported everywhere else in the cell. And they have to do this by crossing the aqueous cytosol. And one of the ways that they do this, and we're coming to realize maybe the primary way, is through these bridge-like lipid transport proteins, which literally act as a bridge across the cytosol. So first, a little bit of introduction. Get rid of this thing. There we go. First, a little introduction. So this image of a cell here, we're probably all very familiar with. And you can see the nucleus in the middle and various organelles, mitochondria, ER, Golgi. And importantly, pretty much all of these organelles and the cell itself are surrounded by membranes. The membranes are highlighted in blue here in this image. And sort of the very basic structure of a membrane is shown on the right here, where you have the charged head groups shown in blue that are facing the cytosol extracellular environment, the aqueous environment. And then you have the hydrophobic acyl tails of the lipids that are pointing inward and are protected from that aqueous environment. The membrane serves many, many super important functions in the cell, such as structural support, providing physical structure to both the cell and the organelles, separating the cell interior and the organelle interior from the environment, and compartmentalizing cellular functions. So you have, for example, energy production happening in one place in the cell, and as well as like regulating entry and exit of various proteins and molecules and whatnot into all of these different compartments. And also, all of these different organelles and the plasma membrane have a very specific lipid composition. And the lipid composition varies from organelle to organelle, and it's important that the lipid composition of all of these different things is maintained in order for the cell to function properly. There are 8 major categories of lipids in the cell. And for example, the plasma membrane is composed primarily of phospholipids, but it's also highly enriched in sterols like cholesterol. And the mitochondria membrane is composed mostly of phospholipids, but it also contains this very specific special phospholipid called cardiolipin. And again, those specific lipid compositions have to be maintained in order for the cell to carry out all of its functions. And there are thousands of different lipid species within a cell. And also, kind of interestingly, those different lipid species vary from organism to organism. So they're different in humans and C. elegans and Drosophila, etc. So lipid transport is also lipid environments and lipid membranes in a cell are also highly dynamic. So they're always changing all the time in response to all of these very fundamental cellular processes that are happening just all the time, most of which we're familiar with, like phagocytosis, otherwise known as cell eating. The cell engulfs things in its extracellular environment. Exocytosis, where the cell releases things into the external environment. Organelle biogenesis, just normal membrane homeostasis, as well as membrane events like temperature adaptation. If you put a cell in a cold environment, for example, it's going to have to modify its membrane in order to survive. And all of these different processes require an input of lipids, and for some of them, a very large input of lipids that has to happen very fast. For phagocytosis, you need to grow a large portion of membrane very quickly in order to engulf something in the extracellular environment. And most of the lipids in a cell are made in the endoplasmic reticulum, shown in purple here. And then they have to get everywhere else in the cell in a very specific way. We now know that this lipid transport process of moving lipids from the endoplasmic reticulum to all of these other different locations primarily happens through the action of lipid transport proteins. And lipid transport proteins exist at what are called membrane contact sites, regions of membranes, regions of the cell where membranes are very close together. So here, for example, we have an ER membrane that's close to another membrane, perhaps the plasma membrane. And if we look back at this slide here, a cell really doesn't look like this. All of the organelles are not separated in space as is implied here. The endoplasmic reticulum is sort of everywhere across the cell, touching all of these different organelles and touching the plasma membrane, which is where all these membrane contact sites come into play. So these lipid transport proteins have been well studied for a really long time, more than 30 years. There are several dozen that are known, and all of them, for the most part, operate via this shuttle mechanism that's shown in the cartoon here, where the lipid transport protein is sometimes, but not always, anchored on one side, and then it grabs a lipid from one side, from one membrane, moves it to the next, and does back and forth with this shuttle mechanism. And usually it'll move anywhere from 1 to 2 lipids at a time. The problem with this is it's slow. How, if a cell needs lots of lipids all at once, is it going to get those lipids through this slow back and forth mechanism, even with, thousands of lipid transport proteins? Well, this question was perhaps answered about 6 years ago when BLTPs were discovered. BLTPs also are found at membrane contact sites, but they're different from other lipid transport proteins because they're proposed to form a physical bridge that bridges the donor and acceptor membranes, the donor being most likely the ER, and the acceptor membrane being the other membrane in the cell, the mitochondria, the plasma membrane, et cetera. And they're shaped sort of like a tube. And the thought is they facilitate bulk lipid transport by just moving large quantities of lipids from one membrane to the other through this hydrophobic tube-like structure. And for that reason, they've been called different names like a hydrophobic conduit, a lipid superhighway, a lipid fire hose, things like that. So there are 5 main BLTP family members, and their names are BLTP1 through BLTP5 officially. But within the field, they've also been called, they're better known by these other names, which is why I've listed them here on the left. VPS13, for example, is much better known than BLTP5. And over here, I'm showing LPD-3 as the C. elegans ortholog of BLTP1, because that's the protein I'm primarily going to be talking about today. But if you look at the right, on the right, these are the alpha fold predicted structures of these 5 family members from C. elegans. And you can see that overall, they have a similar architecture. They have this beta strand backbone that forms sort of a twisting shape, and it's rod-like or tube-like sort of in general shape. It varies in length. BLTP1 is the largest. BLTP is the shortest in all organisms. BLTP1 and 2 have an N-terminal transmembrane helix, whereas the others don't. And ATG2 and VPS13 both have these extra interesting domains, like you can see one down here, the VAP domain, which people really don't know what they do, but they probably help in binding different auxiliary proteins for regulatory purposes. But overall, they have a very similar architecture, and we expect them to have, at a basal level, a similar mechanism of lipid transport. So this is the protein that we are studying. Our primary objective in approaching this project was to understand how does BLTP1 transport lipids? Because that's something that's unknown, is how it's physically moving lipids from one membrane to another membrane. What's the mechanism? And that's also important because BLTP1 is essential for organism survival. If you knock out BLTP1 in humans, mice, or Drosophila, they die. And interestingly, though, if you knock out BLTP1 in C. elegans, the worms actually don't die, they just get very sick. But there's also mutations in BLTP1 that have been implicated in this neurodevelopmental disease called Alkiraia-Kuninskas syndrome. And this disease, which is relatively rare, mostly because most mutations in BLTP1 are lethal, but this disease results in all sorts of physical deformities as well as intellectual disabilities and a whole host of other symptoms which make the disease relatively difficult to diagnose and currently impossible to treat because we don't know the mechanism behind BLTP1 mediated lipid transfer. So therefore, we can't understand how mutations are impacting lipid transfer and begin to think about treatment options and things like that. So being a structural biology lab, we approached this problem with cryo-EM. We wanted to understand what is the cryo-EM structure of BLTP1, and hoping by looking at the structure, we would understand mechanistically how this protein is working in order to design other studies. There have been, before we started this project, other structural studies done of other BLTP proteins, but by and large, they were very challenging and there's essential information missing. And I'm showing a few of those here. You can see for the most part, they resulted in partial or low resolution structures. In all of these different cases, it's obvious that there's this beta strand backbone, as was predicted, that forms the tube-like structure, and there is a cavity in the middle of the protein. But one essential thing that was missing from all of these structures was the interaction between the protein and the lipids, because that's one of the most important things that's going to give us insight into how this protein is functioning. So because these BLTPs are difficult to produce via normal sort of recombinant protein production in heterologous cell lines like bacteria, yeast, HEK cells. And that could be for a lot of reasons that they, well, one, they're very large, so perhaps they had trouble folding or they needed auxiliary proteins. We weren't really sure. So we approached this problem in a different way by purifying the native protein complex of BLTP1 from C. elegans. So C. elegans is a terrific model organism for looking at native protein structures because it's relatively easy to genetically modify, and because it can be grown in great abundance. In a relatively short period of time, one can generate millions and millions of C. elegans from which you can extract your protein of interest. So that's what we did here, is we tagged the native BLTP1 gene in C. elegans with a fluorophore and an affinity tag. And then we did a bunch of validation studies to confirm that, yes, in fact, the protein was still organized, localized to the correct locations in C. elegans, which you can sort of see from this confocal image here. And we also did a series of validation experiments showing that the worms still behaved like wild type, that the lipid transfer dependent processes in C. elegans were not impacted by the addition of this tag. So after we had this validated worm line, we proceeded to grow lots and lots and lots of worms. And first, we tend to grow worms on plates, very large plates, the 15-centimeter ones. And then we move them into liquid culture and grow worms in liquid culture in a similar way as one would grow bacteria in culture, with obviously modifications. And that allows us to get millions of worms from a single liquid culture. And then we freeze the worms for downstream experiments. And for a single experiment, a single cryo-EM experiment or other analytical experiments, we would need somewhere around 50 million worms. And for reference, that's about a 50 mil conical falcon tube that's packed full of transgenic C. elegans. So with these worms, we lysed them by grinding them up, solubilized them, and then went through sort of normal affinity purification, size exclusion chromatography. The difference here being that we're working with very, very small amounts of protein. So we had to adapt these methods to accommodate the tiny amounts of protein we were working with. And then at the end, for cryo-EM experiments and other experiments, we ended up with about 200 nanograms of BLTP1, which is still in the world of structural biology, not a lot of protein, but fortunately is sufficient for cryo-EM if you're using 2-nanometer carbon grids or grids with some other support film. But after establishing this purification method, the first thing that we did was actually not cryo-EM. The first thing that we did was mass spectrometry, because we wanted to know, are there any proteins that are co-purifying with LPD-3, with C. elegans BLTP1 LPD-3? Are we dealing with a complex or a single protein? Because there were, up until this point, no auxiliary proteins that were known to interact with BLTP1, the ortholog from any organism. And we were excited to find that, yes, there were two proteins that were clearly co-purifying with LPD-3. So in this column, you see the peptide spectral counts for the protein isolated from our transgenic worms. And here's our control, which is wild-type worms, to control for non-specific binding during the purification process. And we see very high peptide spectral counts for these two proteins, indicating that they are co-purifying with our LPD-3 protein. And lower peptide spectral counts for a host of other proteins, which I'll talk about a little bit more later at the end. Interestingly, neither of these proteins had ever been studied in any organism. Their function was unknown, which was kind of exciting. And importantly, so we now knew that we were dealing with not just LPD-3, but a complex of LPD-3 plus two auxiliary proteins. So when we put this protein complex on a grid, we collected a little over 8,000 micrographs at PNCC. And then through iterative 2D classification, we ended up with slightly under 1 million particles. Ab initio reconstruction yielded a volume that looked like this. You can see the detergent micelle on one side. This protein has an N-terminal transmembrane domain, so we were expecting a detergent micelle. And then through many rounds of heterogeneous refinement, we eventually ended up with an N-terminal map at 2.7 angstrom resolution. And this is what the map looks like. So in green is LPD-3. You can sort of begin to see the twisting architecture of the tunnel here in the beta strands. And in orange and yellow are auxiliary proteins. And this transparent blob is the detergent micelle. And you can sort of see in some orientations, lipids are shown in light gray, like here. And using this map, we were able to build a model of the complex. And very excitingly, we observed not just auxiliary proteins, but we also saw that the tunnel was packed full of lipid molecules, which is really exciting for us. And since neither of these auxiliary proteins had been characterized in any organism, we named the one in yellow spigot. And we actually didn't know the identity of the one that's colored orange here for quite a while. And its identity was actually later discovered by the Vani and Conaber groups. And the paper is posted on bioRxiv, but it is the C. elegans ortholog of TMEM 170. So the big takeaways from this model here is that one, this is only half of LPD-3. If you were thinking that it looks a little bit short, that's because it is. Two, the tunnel is packed with lipid molecules, like I mentioned, which is very exciting because that gives us some mechanistic insight. And three, LPD-3 is bound to auxiliary proteins. We show two auxiliary proteins here, which are the two we can see in the map, but there's also a third auxiliary protein that we weren't able to observe in the map. And I'll talk about all three of these points. So first, the fact that we're only seeing half of LPD-3. That's especially apparent when you compare it to the AlphaFold predicted model of LPD-3, which is shown on the right here. You can see that we have high resolution information to about here, and then we don't see this C-terminal half of the protein. And we actually did get a low resolution map of the entire thing. And the length of our low resolution map matches the length of the AlphaFold predicted structure. And we're not honestly entirely sure why we're not able to observe the C-terminal half of this protein. It is interesting that the first half is much more compact than the C-terminal half. The linkers on the C-terminal half are a lot longer, a lot floppier, and perhaps that's contributing to some sort of heterogeneity or flexibility, something like that. We would, however, expect this sort of beta strand backbone to be structurally rigid. So we're not sure what's going on, but it's something we're still working on because we would really like to understand how specifically this C-terminal end is interacting with the donor membrane in order to deliver the lipids that are transported. Importantly, though, in our low resolution map, we did see that there's a continuous tunnel from the detergent micelle all the way through the protein, all the way to the end. So that just tells us, helps us confirm that our map is indeed correct. We got to this map by, well, we tried lots of things. First of all, we tried signal subtraction, we tried local refinement using a variety of different masks, and none of those approaches worked. What did end up working the best for us was we used 2D classification to select the particles that had the best full-length features. So they had the best features of this C-terminal half here. And then through ab initio reconstruction, you can see a little bit more of that C-terminal half, and here a little bit more. And then what really helped was recentering the volume. So the CryoSpark really liked to focus on the detergent micelle, probably because it's so bright. So it was shifting the particles than the box. So by recentering all of the particles, we were able to get the map that you saw, which is certainly not 6 angstrom resolution. It's a nominal resolution, but nevertheless. So what about the N-terminal half? Because we can still get a lot of interesting information about how this protein complex is working by looking at the N-terminal half of the complex. So LPD-3 itself, shown in green, is composed of what are called repeating beta groove domains. A repeating beta groove domain, if you look at the bottom corner here, is what's called beta taco structure, where you have these two beta strands meeting in a V. And then that's covered by what we call a loop cap, which is one or more alpha helices in loops that cover the top of that beta taco. And an RBG domain is composed of anywhere from 3 to 6 beta strands. And these RBG domains are modular, and they sort of stack together to form the tunnel. And by adding more RBG domains, you get bridge-like lipid transport proteins of various lengths. So BLTP-3 has only like 6, whereas BLTP-1 has 16. And each RBG domain is rotated a little bit relative to the previous one, which is apparent when you look at it in this view here, so that there's approximately a 270-degree rotation between the first RBG domain and the 7th RBG domain in our structure. And that's why you see that spiraling architecture. And the architecture of an RBG domain on the inside is sort of perfect for solubilizing lipid molecules. If you look at this image down here, everything in gray, all of those gray side chains are hydrophobic amino acids, and which we expected because we knew that this tunnel was solubilizing lipid molecules for transport, so it would need to have a hydrophobic interior. But we also saw a minor amount of charged amino acids. And these charged amino acids were located on the right side of the beta taco and the top of the loop cap. And it was continuous throughout the entire tunnel. So on the right side of the beta taco and the top of the loop cap here was this long string of charged amino acids, so both positive and negative charge, that was continuous throughout the tunnel. And I'll talk about this in a few minutes, but the purpose is to organize the lipid molecules within the tunnel so that you have these charged amino acids interacting with the charged lipid head group, and then you have the rest of the tunnel being hydrophobic for solubilizing the hydrophobic lipid tails. The RBG domains also vary substantially in length. So the first few are quite short, and then as you get towards the middle of the protein, they become quite a bit longer and larger, and it's primarily due to increased length of the loop cap. And so the loop cap becomes much, much longer, and at least in our structure in the first half, for the most part, the loop cap was not floppy and in space. It would wrap around the outside of the tunnel in various ways and was structured. And one of the most exciting things for us, like I've said a few times, is that the tunnel was filled with lipids. So that's especially apparent if you view it like this. And you can see if we slice the tunnel sort of in half, that there's this cavity in the middle, and it's filled with lipids, just completely packed. There was absolutely no empty space within that tunnel. And also, interestingly, you can see that the first half of the tunnel is much narrower than the middle. So in the first half, you have room for anywhere from 1 to 2 rows of lipids, and it widens substantially as you get towards the middle of the tunnel, and we observed up to 5 rows of lipids. And these lipids are arranged in very organized, stacked rows within the tunnel. So looking at this zoomed in image on the bottom, I've numbered the rows here, so you can see all 5 towards the middle of the tunnel. And you can also see these blue and red side chains of those charged amino acids, all of which are positioned right next to the phospholipid head groups. And that's what's helping to organize them. So all of the head groups are pointing in one direction, and all of the tails are pointing in the other direction. And in that way, the organization is actually very similar to a lipid bilayer, because in a lipid bilayer, you would also have all of the heads in one organization and all of the tails pointing in the other. And the density of the lipids within the tunnel, where there were more lipids, like here towards the middle, was also very similar to the packing density within a normal lipid bilayer. So it really looks like there's this little piece of a membrane that's moving through the tunnel. And down here, I'm showing the density for this very first row of lipids, row 1. And so we modeled them all as phosphoethanolamine because, as you can see from the density here, for the most part, it was quite low resolution. The best case scenario, we had this V-shaped structure. And in some cases, we were able to model full lipid tails, but often the lipid tails were truncated due to flexibility. The BLTP proteins are known to transport phospholipids, which is why we modeled them as phosphorethanolamine. But unfortunately, the resolution wasn't good enough for us to be able to identify exactly what lipids are being transported through the tunnel, because that's one of the major outstanding questions in the field of BLTP research, is what proteins are transported by each BLTP under what conditions. So here, showing cross sections of the tunnel, just showing it in different views. You can see the different rows. Near the entrance, there are 2 rows of lipids, and then the tunnel gets narrower to accommodate only a single row of lipids, and then widens again to accommodate 5 rows. And this is due to flattening of the beta taco here, primarily, that accommodates these additional rows of lipids. And if you look below, it's also showing the those charged amino acids that are interacting with the phospholipid head groups. And they're all positioned on the right side of the beta taco and the loop cap. And predictably, the number of these charged amino acids increases as you get more rows of phospholipids in order to further organize them and keep them in that structure. And I just like this image here because it very nicely highlights how you have the acyl tails that are captured in that beta taco and surrounded by hydrophobic amino acids. And then the head group is interacting with these charged amino acids on the top here. So another thing that we noticed is that the tunnel is not completely enclosed. So it's actually decorated by what we called hydration portals, these big openings in the tunnel exterior that enable water molecules from the cytosol to enter and hydrate the lipid head groups. I guess going into this project, I sort of naively thought that the tunnel would be, completely enclosed like a tube. But it actually has these big windows allowing molecules from the cytosol to enter. And our thought is perhaps that this is facilitating lipid flow through the hydration of the head groups. And this is, I think, easiest to see in cross sections here, where you can see that some regions of the tunnel are completely enclosed, like here, whereas other regions, you have these portals where there's this big opening that's positioned immediately above the phospholipid head group, sort of supporting this idea that water molecules can enter from the cytosol, hydrate the lipid head group, and potentially facilitate flow through the tunnel, perhaps preventing stagnation or just facilitating the energetics of the process. And again, this is also really important. This is also really interesting because this is exactly how the lipids would be in their native environment in a lipid bilayer. Is the head groups would be exposed to the aqueous cytosol, and the tails would be protected in the interior of the membrane environment. So again, it really just looks like a little piece of a membrane bilayer that's moving through the lipid transport tunnel. So what about the auxiliary proteins? I said in the beginning that we did mass spectrometry, and we identified 2 different auxiliary proteins that were co-purifying with LPD-3, and those were spigot, which is in yellow here, and we saw in our cryo-EM structure, our cryo-EM map, and then LTAP2, which we named lipid transfer auxiliary protein 2, which we didn't give it a fun name because we couldn't actually see it in the cryo-EM map. We're thinking that perhaps LTAP2 is binding somewhere on the C-terminal half of LPD-3, which is why it wasn't visible. But we did see density in the transmembrane domain for another protein that was composed of 3 transmembrane helices. And at first, we had no idea what this protein was. But later, its identity was determined by the Conabert and Vani groups, which showed that it is TMEM170. They also did some nice studies to show exactly what its function is in lipid transport that I'll talk about a little bit later. So looking at these auxiliary proteins, our main questions were, 1, what are they doing? What is their role in lipid transport? And 2, are they conserved members of BLTP1 complexes? So here we're looking at the C. elegans complex, and we want to know, are these auxiliary proteins also interacting with BLTP1 in Drosophila in humans, indicating that they are, in fact, conserved members of these complexes and playing a role in BLTP1 mediated transport. So to answer these questions, we worked with Dr. Yoon-Sik Kang at the University of Colorado, and we did a series of assays to show that if we knock down these auxiliary proteins in various organisms or cell lines, what is the effect on lipid transfer dependent processes? So if we knock down spigot and we see an impact on processes that are dependent on lipid transfer, it suggests at least that these proteins are important for lipid transport. So the first thing that we did was knock down spigot in C. elegans and look at actin organization. So actin organization is dependent upon proper lipid distribution, and a normal apical actin membrane looks like this, where you see these two sort of straight lines. And then you knock down LPD-3, and it's a complete disorganized mess, and the worms are very sick. Knock down spigot, and it's also a complete mess, indicating that spigot is important for lipid transfer in C. elegans. We did a similar things in flies. So here we have a fly larva, and this is the brain of the fly larva. And that's what we're looking at here is this bottom half over here, and highlighted are the neurons. And in flies and in other organisms, as they develop, there's this stage where there's a lot of neuronal death. And then those neurons have to be cleaned up by astrocytes, that neuronal debris of the dead neurons and synapses and whatnot. And under normal conditions in a control, after 12 hours, all of those debris are completely cleaned up by astrocytes. And the astrocytes are, they engulf all of these different debris, which is a phagocytic process. But when you knock down BLTP1 or you knock down spigot, there's a lot of debris remaining, indicating that phagocytosis is not happening properly. So perhaps, which is a very lipid transfer dependent process. And then lastly, we showed in HeLa cells that if you knock down spigot, you get a collapse of these endoplasmic reticulum plasma membrane contact sites. So in a normal cell, if you look at the membrane contact sites, they look like this, sort of around the edge, highly organized. But when you knock down spigot or knock down BLTP1, we saw a collapse of those ERPM contact sites, and the cells were very sick. Most of them actually died. But in the surviving cells, we did not observe any more ERPM contact sites, indicating that perhaps spigot is important for stability of ERPM contact sites. So all of this data together suggested to us that yes, spigot is in fact a conserved member of BLTP1 complexes. And what is its role in lipid transport? That's still unclear to us. But looking at the cryo-EM map, we can see that LPD-3 and spigot interact extensively. So spigot interacts with the transmembrane domain of LPD-3, and then it sort of wraps around the outside of the tunnel in the cytoplasm and terminates in this 4-helix bundle that's embedded in the side of the tunnel. And most of these interactions are electrostatic, but it's a very extensive interactions that's keeping spigot attached to LPD-3. It could be necessary for stability, could be necessary for localization to the ER. We're not really sure at this point, but it is definitely essential. LTAP2 is a bit more of a mystery. So we did a similar set of experiments where we knocked down LTAP2 and looked at actin organization, and then looked at other features of C. elegans development and cold tolerance, both of which are dependent on lipid transfer. And we saw it's not important for actin organization at all. Knock down LTAP2, and actin looks just fine. But if you look at the developmental rate in C. elegans is a little bit impaired when you knock down LTAP2, but the cold tolerance of the worms is completely fine. So it's kind of unclear here. However, if you knock down LTAP2 in flies, we do see this impaired phagocytosis, indicating that it might be important for BLTP1 mediated lipid transport. So at this point, maybe additional studies are required. And since we can't see LTAP2 in the cryo-EM map, we really have no idea what its role in lipid transport is, something we're also investigating. But the C. elegans ortholog of TMEM170 is the one that we have the best idea of what's going on here. So the Vani and Conaber groups in this preprint that's currently on bioRxiv, they showed that TMEM170 is a scramblase, and it is a conserved member of BLTP1 complexes. So it's composed of these 3 transmembrane helices, and they showed, you can see this gray line sort of snaking up through the middle, that it's a lipid scramblase, and this is the lipid scrambling pathway through the membrane that leads right up through the center of those 3 transmembrane helices. And they showed this through a series of computational experiments and also in vivo experiments in yeast. And looking at the model, if we sort of very roughly map that lipid scrambling pathway on the model, you can see that it leads right to the right-hand side of the entrance to the BLTP1 tunnel. And that's right where those phospholipid head groups are organized by that line of ionizable amino acids that line the interior. So I think that's pretty intriguing because to me, it at least suggests that this lipid scrambling pathway might be feeding phospholipids into the tunnel and facilitating organization by feeding them in on that right-hand side of the beta taco. So what about all of these other subunits? When we did our initial mass spectrometry experiment, we identified LTAP2 and spigot as definitely proteins that are co-purifying with LPD-3, but there were also all of these other proteins which had much lower but decent peptide spectral counts. And we thought, well, maybe they are weakly bound proteins, or they're regulatory proteins or adapter proteins, things like that aren't part of the core complex, but are indeed bound to LPD-3 some portion of the time. So to investigate this possibility, UNSIC did the same astrocyte phagocytosis assays in fly larvae and showed that, yes, when you knock down these proteins, there is a phagocytosis defect in most cases, indicating that most of these proteins could potentially be involved in LPD-3 mediated lipid transport, which is kind of interesting. So this is another thing that we're interested in investigating is what is the role of these proteins, if anything, in LPD-3 function. So finally, this cartoon is a quick summary of what we learned from our cryo-EM structure. We learned that phospholipids are organized in the tunnel via this track of ionizable residues that it runs all the way from the ER membrane through the entire length of the tunnel that we can see. It interacts with the head groups in order to organize them into rows. We also saw these big hydration portals in the tunnel that we believe is allowing water molecules to enter the tunnel and hydrate the lipid head groups, potentially facilitating flow. And we showed we and others showed that spigot and TMEM170 are conserved members of BLTP1 complexes and possibly LTAP2. It's just a little bit unclear at this point. But there are still many outstanding questions. There's still so many things that we don't know about how these bridge-like lipid transport proteins are working. We're still focused on BLTP1, and we want to know where is LTAP2. So we think it has to be binding to the C-terminal half of the protein. And we want to know what its role is and where it's binding. How does the C-terminal half of LPD-3 attach to the acceptor membrane in order to deliver lipids? It doesn't have any amphipathic helices or obvious hydrophobic patches that would allow it to interact with the acceptor membrane. So there's this thought that perhaps it's binding to different auxiliary proteins, adapter proteins, in order to anchor it on the other end, but we're not really sure. What types of lipids are transported and what is the mechanism for selectivity? It's relatively well known that BLTPs are transporting phospholipids. But since the membranes of all of these different organelles varies so much and is so important for the integrity of the cell, it's expected that there is some sort of mechanism for selectivity. We just don't know what it is yet. And also, what is driving lipid transport is one of the biggest questions. As far as we know, this is not an ATP-dependent process. So what is driving lipids from the donor to the acceptor membrane? Is it purely a gradient process where there's more lipids on one side that's pushing them forward? Is lipid transport reversible? Can lipids go in the other direction? We don't know. All things that we're curious about. And finally, I just want to thank everybody in my lab. Hannah, Jolene, and Sarah are grad students. Sophia and Avery are undergraduates. Yunsik, who did just about all of the in vivo studies that you saw today. PNCC, where we collected all of our cryo-EM data and our funding sources. And I want to note that postdoc positions are available in case there's anyone interested. Thank you. I'd be happy to take questions.
Thank you, Sarah, for a great talk. And you do have time for a few questions. And I do see a question from an enthusiastic attendee, and they ask, what about specificity of lipid transport? Do we understand if one BLTP can transport one type of lipid or multiple?
Yeah, we don't understand that. It's thought that one BLTP can transport multiple different types of lipids, but I guess the primary assumption without a lot of data is that one BLTP can sort of transport a particular class of lipids that are perhaps similar in their chemical structure.
I see. Another question is about how does lipid translocation occur between the occluded and hydrated pockets?
How does what occur?
The lipid transport, translocation. How does the lipid translocation occur between the occluded and hydrated pockets?
Oh, yeah. So I guess that would rely on the driving force, just a constant movement from the donor membrane to the acceptor membrane, sort of moving it between those hydrated pockets and non-hydrated pockets, just a constant flow. And I think that's why we were able to see the lipid densities in our cryo-EM map, is because when we isolated this protein, we sort of removed it from any driving force. So the lipids became stagnated within the tunnel.
Sounds good. Okay, one more question. So when you dock the AlphaFold model in the low resolution complete map, do you see extra density that could be the missing protein?
No, we don't. And interestingly, when you dock the AlphaFold map model in the low resolution map, it doesn't fit perfectly. So it's the angle is different. If the AlphaFold model looks like this, then our cryo-EM map looks like this. The C-terminal half sort of comes off at a different angle, and we don't see extra density.
One more question. Is there a sensory mechanism on how the lipid need is sensed and responded?
Yeah, that is another big question in the field. So what are the regulatory mechanisms at play so that the place where lipids are needed have lipids delivered, and not only that, but the correct lipids are delivered to that place? No, that's totally unknown.
I see. I don't see any other question, but I do.
I have another question.
Mhm.
I was wondering, what methods are you trying or to help stabilize the C-terminus in your cryopreps? Are you looking at cross-linking? Are you looking at what type of methods are you approaching or trying?
Yeah, initially, cross-linking is something we're trying. The other things we're trying initially are just really fundamental, I guess. Different grid surfaces, so glow discharging under different conditions. Trying GO versus 2 nanometer carbon. And if those things fail, then my primary thought was to some method to try and add weight to the C-terminus so that it's more easily identifiable by processing programs. But yeah.
I recently saw another paper where they increased the viscosity of their solution by adding a lot of background PEG to make mobile things slow down and stop wiggling as much, which is another interesting approach.
Huh. I'll look into that. Yeah, Michael.
Yeah, you might also try a method developed in at Stanford SLAC here about the Gaussian mixture model, which can do a focused classification and put more weight on certain parts of the certain areas of the structure, but still having a weight that is on the obvious part of the structure that says, oh yeah, we know that it's there, but now we want to concentrate more on the parts that are lower resolution and or more flexible. The Gaussian mixture model is that method that's developed by Muyan Chen here.
It's another data processing analysis method to look at variability.
Okay, yeah, thank you. I hadn't heard of that one. I'll definitely look into it.
Mm-hmm.
And I have a cryo-EM question as well. This particle looks really hard to pick. Did you struggle with particle picking, or was it more straightforward than I'm thinking it might have been?
Yeah, particle picking actually was very straightforward. The primary thing we struggled with was we had to have a really big, long particle. So we had to use a really large box size, which just slowed things down a lot. We had a lot of failing jobs initially until we figured things out. But yeah, particle picking was easy.
And did you have preferred orientation issues? Like, I'd imagine this thing, how many top views do you get?
Yeah, none. We didn't get
Okay.
any top down or bottom up views. It was pretty much all laying on its side, and then you can picture it sort of rotating on its side like that.
Yeah.
Yeah.
That's usually good
But we
enough.
didn't have any preferred orientation of it on any of the different side views.
That's
any tilted data collection or all at zero tilt?
All at zero. You think tilted would help in any way?
I think tilting would get you some more oblique views of the top and bottom ends of the structure.
Okay.
Yeah, whether or not that gets you to more biological information, it's hard to say. But yeah, that because you might imagine you need you need to be in like a thicker ice, but obviously that's bad for imaging. So I'm actually curious about Nathan's suggestion about the PEG, right? It's going to hurt you in some ways, but may help you in other ways.
Yeah.
As long as you have azimuthal views of 360 degrees this way, that provides all the information you need. It's a little bit, it helps if you have the views exactly towards you. I mean, up and down, but you don't really need it. You've filled in reciprocal space completely if you've done that.
Yeah. Yeah. Yeah, we're hoping just by changing different sample conditions and grid surface conditions that we can try and get that C-terminus to be more, because we're not even sure if it is mobile. We're not sure what's going on. Yeah. It seems like it should be structurally rigid looking at the AlphaFold predicted structure.
All right, well, we're coming up on the hour. I see we had one more question that came in. We'll stick around to answer that. But for anyone who has to run off for their next meeting, thank you for joining us today. We wish everyone well in your cryo-EM endeavors, and the national centers are here to help us with your science. So let us know how we can help. We hope to see you again, and we'll get to that one last question before we call it a day.
All right, so it says, thank you. Thanks for sharing, of course, this, they think. So did you try any signal subtraction of other focus refinement methods for this work?
Yes, we did. We tried signal subtraction of varying lengths of the N-terminus, the whole thing, just the micelle, various pieces, which didn't work at all. And we also tried local refinement using a whole bunch of various sizes of masks on that half. And that didn't work either.
Great. Thanks for sticking around for that last question.